Chapter 11, Clicker Questions for Endoplasmic Reticulum and Golgi apparatus protein processing and sorting. The endoplasmic reticulum, Golgi, and vesicle secretory pathway is used by cells to synthesize and transfer proteins to any compartment that requires a crossing membrane. This is false. Other than proteins, the Golgi, and vesicle secretory pathway is also very important in lipid metabolism, as well as storage for calcium ions. Number 2, signal sequences that target proteins to the endoplasmic reticulum are usually deficient in arginine, glutamine, and lysine because these amino acids have blank side groups. Is it polar, bulky, nonpolar, or hydrophobic? The answer is A, polar. Since the majority of the signal sequences will have 7 amino acids that are going to be hydrophobic, not hydrophilic. Number 3, what role do translocans play in inserting proteins into the ER membrane? A, they allow parallel alpha helices to be inserted from both sides of the ER membrane. B, they allow the insertion of alpha helices from the amino terminus on the cytoplasmic side. C, they allow ribosomes to cross into the ER lumen, where they can synthesize alpha helices from the inside out. Or D, they allow mRNA to cross into the ER lumen, where it can be translated by ribosomes on that side of the membrane. The answer is B. They allow the insertion of alpha helices from the amino terminus on the cytoplasmic side. So the important thing to remember is inserted in through the cytoplasmic side. 4. What is often the first step in the processing of newly synthesized ER proteins? A, chaperone-mediated protein folding. B, the addition of sugar molecules or glycosylation. C, proteolytic removal of the signal sequence. Or D, disulfide bond formation. The answer is C, proteolytic removal of the signal sequence by the signal peptidase. 5. How does the ERAD or ER associated degradation system recognize misfolded proteins? A, misfolded proteins are blocked from entering the lumen of the ER system by the cytoplasmic ribosomes. B, the reducing environment of the ER lumen causes misfolded proteins to aggregate. C, misfolded proteins have their signal sequence buried in the protein's interior, so it can't be removed. Or D, protein processing enzymes look for hydrophobic regions that will be shielded if the protein were folded correctly. The answer is D. Protein processing enzymes look for hydrophobic regions that will be shielded if the proteins were folded correctly. So very simply, the enzyme UGGT, if you remember, is UDP, glucose glycoprotein, glycosyl transferase. Very simply, will keep adding glucose back into the oligosaccharide and basically look to see if it's going to be bound again by the calreticulin or the calnexin. Which will help kind of refold the protein. Upon unsuccessful protein folding, then it will be ubiquitinated and therefore degraded. What is the fate of Golgi resident proteins in the operation of the Golgi complex? A, they are transferred to the trans-Golgi network and lost from the Golgi complex. B, they are recycled and returned to Golgi complex by vesicles pinching off the sides of the stack. C, they are anchored with individual Golgi cisternae, where they remain and are degraded. Or D, they remain attached to the proteins they are modifying and are secreted from the cell. The answer is B. They are recycled and returned to the Golgi complex by vesicles pinching off the sides of the stack. So depending on whether you follow either the cisternal maturation model where each cisternae will gradually move from the cis to the trans face, or the stable cisternae model where you have vesicles will go from one sac to the next, usually in the cis to trans, but in this case from the trans back to the cis, the resident proteins are simply recycled and returned back to an earlier partition or a closer cis sac of the Golgi complex. 7. Many viral coproteins are glycosylated. Why would this be advantageous? My apologies for the error here. A, glycosylation of viral coproteins would make it easier for the virus to evade the host cell's immune system. B, glycosylation of viral coproteins would aid in trafficking of viral particles from one cell to the next. C, glycosylation of viral coproteins would allow the viral particles to remain in the cytoplasm during host cell mitosis. Or D, glycosylation of viral coproteins would aid in the Golgi-mediated export of mature viral particles. A, glycosylation of viral coproteins would make it easier for the virus to evade the host cell's immune system, but the other side effect too is, especially with different types of viruses that spike proteins such as SARS-CoV-2 as well as COVID-19, they basically allow these particular viruses to bind to the target receptors and actually enter the cells and basically take over the cell's metabolism to produce more of the individual virus proteins. 8. What function of plant Golgi complexes is unique to plant cells? A, they participate in photosynthesis, which is unique to plants. B, they participate in maintaining proper intercellular water homeostasis. C, they participate in the packaging and processing of proteins for secretion. Or D, they participate in cell wall formation and animal cells lack cell walls? The answer is D, they participate in cell wall formation and animal cells lack cell walls. So very simply, there really is only two possible answers through here. So very simply, either A or D, but A is simply not true, since the participation photosynthesis is actually done in the chloroplast, not done in the Golgi complexes. So that makes the answer false. So D is your best answer. Number 9. What is the role of secretory granules in protein export from the Golgi? A. They transport proteins destined for export directly from the trans-Golgi network to the plasma membrane. B. They recycle membrane and proteins from TGN back to earlier compartments in the Golgi complex. C. They fuse with late endosomes to form a lysosome, which is used to digest food. Or D. They store condensed proteins until an external signal calls for the release from the cell. Here the answer is D. They store condensed proteins until an external signal calls, usually a hormone, for the release from the cell. So in this case, you have different stages of secretory granules. Basically, they would start out as immature once they pinch off the Golgi apparatus, until they fully mature, until it actually is released by the external signal, where it's going to fuse the plasma membrane and dump the contents either into the extracellular space or into some type of blood vessel. Number 10. What makes epithelial cells unique in terms of protein trafficking? A. They're involved in the active uptake of nutrients and must have receptors over their entire surface. B. They are polarized, so different proteins must be trafficked to different regions in the same cell. C. They are secretory, so all of the proteins must be trafficked to the apical surface for export. Or D. They must be able to distinguish between proteins that have been glycosylated and proteins attached to anchoring lipids. The answer is B. Epithelial cells are polarized, so different proteins can traffic different regions of the same cell. So very simply, you can have either different types of secretory proteins either sent to the apical surface, which will actually face an open space, whether it be a lumen or the outside surface, or towards the basal area, which could basically shunt different types of nutrients into the neighboring blood vessel, or possibly go out to the periphery, adjacent to other epithelial cells like anchoring proteins, basically help stabilize the entire area. 11. True or false? After their formation, vesicles diffuse from the trans-Golgi network, or TGN, to the plasma membrane. This is actually false. There are multiple destinations for vesicles once they diffuse from the trans-Golgi network. They can either fuse the plasma membrane, or they can be secreted, or possibly fusing with lysosomes, different vesicles to form other types of membrane-bound organelles, or simply placed back into the ER, or possibly back in different cisternae of the Golgi. 12. What would happen if a cell could not produce dynamin? A. COP1 vesicles, or coprotein 1 vesicles, could perform but could not move in the retrograde direction and recycle Golgi resident proteins. B. COP2 vesicles would move in the retrograde direction instead of an anterograde direction. C. Clathrin-coated vesicles could form but could not be released from the surface of the ER membrane. Or D. Clathrin-coated vesicles could form and be trafficked but could not fuse with the plasma membrane. The answer is C. Clathrin-coated vesicles could form but could not be released from the surface of the ER membrane. Very simply, the whole function for a dynamin is to form a dynamin ring around the budding vesicle to basically pinch itself off from the surface of the ER membrane, and therefore to be transported through the different types of filaments of the cytoskeleton of the cell to its particular destination. COP1 coat proteins are needed to A. Scavenge and return Golgi resident proteins, B. Guide vesicles to the late endosome, C. Traffic vesicles to the plasma membrane, or D. Move vesicles from the ER to the endoplasmic reticulum Golgi intermediate compartment, or the ERGIC. Answer is A. Scavenge and return Golgi resident proteins. So if you remember back from the chapters, COP1 is going to actually bring things backward through the ERGIC complex. Very similar, they're going to return the resident proteins to early compartments of the secretory pathway, whereas COP2 coated vesicles are going to carry the proteins from the ER to the ERGIC and then also the Golgi apparatus, and then budding off into its individual destinations. 14. Vesicle fusion with the correct target membrane requires that A. Clathrin binds to the target membrane and squeezes the vesicle contents to the cell exterior. B. A Rab protein on the vesicle binds to a specific tethering factor in the target membrane. C. Either COP1 or COP2 must be present to aid in target membrane recognition. Or D. Snares, otherwise known as SNAP receptors, on the vesicle and target membranes interact and drive fusion of the two membranes. The answer is twofold here. We talk about vesicle fusion. Very simply, it's a two-step process. First, the Rab protein on the vesicles has to bind to specific tethering factor in the target membrane, and the snares on the vesicle and target membranes interact and drive fusion of the two membranes. So it's going to be B and D. So very simply, the Rab protein will recognize a particular vesicle, which is target membrane, and then the snares very simply will actually have almost like a zipping effect onto the vesicle and target membranes together. So very simply, we'll add the fusion of the two distinct vesicle as well as the target membrane of the particular organelle, and leads to further fusion of the two membranes. 15. Lysosomes contain acid hydrolases. What do hydrolases do? A. They split water into hydrogen and oxygen gases. B. They perform a reaction that is opposite of a dehydration synthesis reaction. C. They increase the permeability of lysosomal membranes to allow for proton-ion uptake. Or D. They cleave covalent bonds and add hydrogen to one exposed bond and hydroxyl to the other. The answer is twofold, B and D. So lysosomes, having the acid hydrolases, very simply, are there for breaking down different proteins and other types of macromolecules. So in this case, letter B, so they perform the reaction that's the opposite of dehydration synthesis. Synthesis is the buildup, and we're looking at the catabolism or the breakdown of certain types of macronutrients, in this case proteins. And as they do this by cleaving the covalent bonds within amino acids and other macromolecules, where they add hydrogen to one exposed bond and hydroxyl to the other. Number 16, lysosomes are unique in that they A. are derived from the Golgi apparatus, B. are only found in animal cells, C. have a low internal pH of 5.0, or D. are formed by the fusion of two separate organelles. Letter is D. Lysosomes are formed by the fusion of two separate organelles. So this is paired with endocytosis, as a cell tries to draw in different types of nutrients from the aqueous surface, and it leads to fusion with transfer vesicles that will butt off the Golgi apparatus. When the two fuse, basically the transfer vesicle with the acid hydrolases will fuse with the contents of the endosome to form the lysosome. Basically, we'll have the different types of reactions to break down those macromolecules into smaller amino acids or other monomers for the cell to use. Number 17, only macrophages are affected in type 1 Gaucher disease. Why is this? A. The main function of a macrophage is phagocytosis. B. Macrophages are dependent on the nutrients released by autophagy. C. Autophagy requires the cells be supplied with an external food source. Or D. Late endosomes must fuse with the macrophage to form a lysosome. The answer is A. The main function of a macrophage is phagocytosis. In Gaucher disease, as we know, macrophages are going to be pretty much the only cell that's affected. And the main function for a macrophage is to basically break down any kind of macromolecule into smaller, reutilizable monomers. So very simply, if the lysosome cannot break certain things down, those particular substances can build up in a particular cell and either, at the very least, disrupt the cell with its normal function or actually cause the cell to necrose, which can naturally build up a certain amount of cell necrosis, can cause different types of organ damage. For 18, autophagy requires the fusion of blank with a blank. Is it A, phagolysosome and mitochondrion, B, transfer vesicle and late endosome, C, lysosome and autophagosome, or D, tethering factor and snare? The answer is C. For autophagy to be performed to be the fusion of both a lysosome and also an autophagosome. So this method is used to either recycle either damaged or old either organelles or other types of cell constituents that need to be broken down and recycled. Very simply, the lysosome will need to basically fuse with the double membrane autophagosome of the organelle or other cell parts to be recycled. Once that fusion occurs, the lysosome will do its job to catabolize those particular parts into recyclable monomers and other smaller molecules for the cell to reuse.